Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to detect viral sequences in regular RNA sequencing data. So why do we need to identify viral sequences? Well, when we extract RNAs from our tissues, we inevitably introduce foreign RNAs from our environments. For example, from human tissues, mice, bacteria, or viruses. Among them, some are contaminations, and some are functional. For example, proteins from HPV viruses can disrupt cellular functions and contribute to carcinogenesis. Therefore, in many cancer studies, viral detection is crucial. In this tutorial, I'm going to show you two ways to detect viral sequences. The first tool we're going to use is called EsViritu. As usual, our first step is to set up our conda environment and install all essential packages. And then we set up our directory for databases. And then we download the virus database using wget with the URL from Xanadu. The download file is zipped file, so we have to unzip the file using tar zxvf. I'm missing a z over here. And then we remove the zipped package. And then we add our database to our conda environment using conda env config vars set. Once everything's set, we can directly run the package. The process is very straightforward. We create our output folder and call EsViritu. Specify the input, that's our FASTQ files, R1 and R2. Specify the name of our sample. Specify the output directory we just created here. Specify the thread we're going to use. Specify the paired, because we have a pair and sequencing data. -Q and -F means we want to QC and filter our FASTQ files. And that's it. The results will be stored in an HTML file and a text file. The HTML file is the interactive file. Hovering your cursor on the file will show the details. So here's the output format. The first column shows you which viruses you have identified. And percent covered, that means 99% of our reads map to the HPV16. The RPKMF is the normalized expression, and reads align is the raw counts. And coverage shows where do the reads cover the genome. And this tool can also work on whole genome sequencing or whole exome sequencing. So it's versatile. The second tool I'm going to introduce is called VIRTUS2. As usual, we introduce our conda environment, install all the essential packages, and download the VIRTUS2 directly from GitHub using git clone plus the URL. This tool relies on Docker, but on HPC, Docker is not allowed due to the security issues. So we'll have to rely on another tool called Singularity. So how do I do that? So once we download the VIRTUS2 on HPC, go to the folder, find the two files, the create index file and the virtus.pe file, edit the file using the text editor, replace the first row with this. Specify the Singularity. Tell the system we're going to use Singularity instead of Docker. So what is Docker? Remember the conda environment I introduced in my first tutorial. They are environments, they are independent environments with a set of tools for a task. For example, you create an environment for RNA sequencing data with all the tools in it, so you can use it independently. Well, Docker goes further. It not only packs the entire environment into a container, it also includes all the operating system in the container. So everything, tools, software, environment, and operating system into one container, and they ship the container to the end users. So as the end user, we just need to download the container and use it directly without setting up the environment or installing anything. So it's very handy. But it requires the root access. That's why it's not safe on HPC. So Singularity is the same thing, but designed for HPC. So if you are using HPC of your institution, you have to use Singularity. Alright, so once everything is set, we can download our database. So for this too, we need two databases. One for viruses, the other one for human, because we're having a human data set. Actually, this two only works for human data set and only work for RNA sequence data. So it's quite limited. So we can download and install the index directly using a function from the package. It's called create index.cwl, and we specify URL here. Just copy it and specify the output name and a folder over here. And once it's set, it will start building the index by itself. So once it's built, the rest is simple. So call the function virtus.p.cwl because we have a pair end sequencing data, and specify the input FASTQ1, FASTQ2, specify the index we just made, the human index, specify the virus database we just created, and specify the name of our sample, and specify the thread we're going to use. So the results are going to be stored in a text table. And let's look at the table. So the first one is HPV16, consistent with the previous two. And look at the number reads. This count is 8,000. Remember our first two? It mapped like 10,000 reads to the HPV16 genome. So it's very close. So here shows 98% of the reads mapped to HPV16 genome. In our previous two, 99% of reads mapped to HPV genome. So it's very close. But remember, this two is very limited. It only works on human RNA sequencing data. So choose the two depends on your needs. So that's all I have for today's tutorial. And for more information and details, just go to my website, ngs101.com, copy the code, and try them on your own. Thanks for watching, and subscribe if you like it. See you next video.